Once the bacteria have gotten into the deeper layers of tissue, it then has to avoid the host immune system. One way that the bacteria can avoid the host immune system is with a capsule. So remember that capsule is that glycocalyx layer, that slippery layer around the bacteria. This can help the bacteria avoid the host immune system by preventing phagocytic cells, these white blood cells, from grabbing onto the bacteria and eating them. So this can just sort of make the cells slippery, prevent them from grabbing hold. So capsules can help bacteria avoid phagocytosis, and that prevents them from being destroyed by the immune system. They can also be used to accumulate proteases that can break down antibodies. Another way that bacteria, gram-negative bacteria particularly, can avoid the host immune system is with the O antigen. So remember, LPS is made up of the lipid A and the O antigen. So the O antigen is what's called the antigenic portion. So it's what your immune system actually recognizes to identify one bacteria and to tell them apart. This varies widely between bacteria, which makes it difficult for the immune system to recognize even closely related bacterial strains, which helps them to avoid the immune response. There are even some types of bacteria that can change their O antigen after they've started to infect you, meaning that they're able to essentially reset your immune system and prevent it from recognizing them. These O antigens can also prevent the binding of an antimicrobial protein called complement, and it can also help prevent phagocytosis. Bacterial species with really long O antigens are sometimes called smooth, whereas ones with shorter O antigens are sometimes called rough. There are also exoenzymes that can help avoid the immune response. One exoenzyme that helps to avoid the immune response is what's called coagulase. So what coagulase does is it converts a protein called fibrinogen in your blood into fibrin. And what this does is it forms these clots. And while the bacteria are inside of these protective clots, they're essentially masked from the host's immune system. They work like a cloaking device. So the bacteria can reproduce freely inside of there, hidden from your immune cells, and then once enough bacteria have built up inside of the clot, they release these enzymes called kinases. And the kinases cause the clot to break down, and then a lot of bacteria get released into the system, and they're more likely to cause a successful infection. We can test for coagulase using the coagulase test, where rabbit serum is added to the bacteria, and then we look for the bacteria causing the serum to clump up or to form a clot. There are also toxins that can help the bacteria to avoid the host's immune system. One is what's called PVL, or panton-valentine leukocidin. This is a membrane-disrupting toxin produced by S. aureus that's a leukocidin. So it's actually going to target white blood cells, part of your body's immune response. So it's going to form these pores in the membrane and trigger the white blood cells to release these damaging enzymes that they would normally use to fight off pathogenic invaders. These damaging enzymes cause edema, swelling, erythema, redness, and necrosis or tissue death, and this can actually lead to these skin lesions that help the bacteria to spread from host to host, while also killing off the white blood cells, preventing a normal immune response. Another toxin that can help protect bacteria from a normal immune response that's also made by certain strains of S. aureus is toxic shock syndrome toxin. This is a super antigen toxin. What it does is it causes a massive, non-specific activation of immune cells called T cells. So during a normal immune response, only certain T cells get activated. Less than 1% of your T cells become active. With the presence of toxic shock syndrome toxin, up to 20% of T cells can become active and cause the release of large amounts of cytokines, leading to what's called a cytokine storm. And this cytokine storm can cause decreased blood pressure, shock, organ failure, and death, aka toxic shock syndrome. And this is going to prevent the body from having an effective, specific immune response. So remember, for pathogenesis, we're going to have exposure. The pathogens are going to enter the body through a portal of entry. Then adhesion. Once they're in there, they have to stick around by attaching to the body cells. Then invasion, where they're going to damage tissue to get deeper into the body and avoid the host defenses. And then they can cause infection, where they're able to reproduce freely and cause disease, that abnormal state with those recognizable signs and symptoms.